Hello everybody, my name is Bowen and I am a second year PhD student in UT Health San Antonio. My program is BME and I'm on the imaging track. So for this class, molecular biophysics, I want to give a talk about this technology I have been researching for this semester. It's called SeqFISH. When we actually look at the test book about we typically see the DNA have this double helix, and then when you zoom out, we see that it's wrapped around the histones. Eventually, you got this chromosome looking like things and actually happen in the metaphase. So most of this time, the chromosome doesn't look like this. Whenever you look into the microscope and you stain the cells with dye, which dye is used in visualizing nuclear DNA in both living and fake cells, you see that there are complex nuclear stains in the nucleus, and you can see dye dim stains. It's actually hard for us to wrap our head around to link those two images together, and a very oddly structured visit with something that look relative difference among different cells. So, of course, nuclear isn't just made of just DNA. In fact, there are many nuclear compartments, and this compartment are made of different type of proteins. And these things we call them compartments for nuclear bodies, and also you can call them domains. And so on, you can see different structures. There are nuclear sparkles and PML bodies and nuclear pore complex, all those together. It is important to look at both the proteins as well as the nuclear acid. I'm going to show you that just looking at each component separately could lead to a very misleading picture of the nucleus. When you have all these measurements in the same cell, you can arrive at a very clear and precisely, relatively more precise interaction between the protein part and DNA part, as well as overall structure, make a lot more sense in the single cells level. So what people have done in the past decades of trying to measure the DNA interaction and the conformation in nuclear is developing this assay called Hi-C technique. So Hi-C has first been applied from the cell lines level. So you culture the cells and you can accumulate the cells to a certain numbers and then we use formaldehyde and other ways to froze the cells, the DNA interactions, and then we digest them, keep the DNA, the interaction level, and then we generate the DNA library and we sequence them. In that way, we align back to the chromosomes in this, and then we can get generate this contact map of which loci is in contact with which other one, and through the computation model, we can reconstruct what nuclear look like. And of course, this is require a bunch of computational resource and power, and this is usually done on the bulk data. But most recently, there has been a lot of improvement of this technology. Now you can apply Hi-Cs on the tissues level. There are commercial kits to support that, and in some labs right now, and also in some I think there are also some commercial kits for single cell Hi-Cs to be used as well. So for example, this is one of the pioneers lab in this field in Dr. Sher's lab at the Harvard University. They did Hi-C in the human lymphoblastoid cells line, and also the human primary blood cells, which is in here I show the GM 12878 cells. And you can generate those really nice chromosome pictures, and then how they interact with each other, how they fold into the nucleus. So, but on the other side, there's a different way we can achieve that is using the imaging approach. People traditionally have done DNA FISH. So the DNA FISH this is how they work. A DNA probe which on the top you can see is a small piece of the purified DNA tag with the fluorescent dye, and the fluorescent is labeled probe find and then bind to the matching sequence within the set of chromosome with a special microscope. At the beginning, they use expansion microscope. The chromosome and the subchromosome location where the fluorescent probe bound can be seen. With different DNA probes, you can design and you can cover the whole chromosome. In this way, you can actually paint the chromosome, right? But we want to be able to look at the genome at the level of thousands of loci and for potentially more. So how could we do that? It's not just one or a few of dots, it's thousands of dots. So of course we're going to have a lot of issues. The two major challenge are multiplexing and the optical crowding. So one of the major limit of the microscope is that there's only limited available colors. So on the typical confocal microscope, we have around 4 or 5 different colors you can neatly distinguish. And however, when you actually look at the really dense dot of the 4 or 5 color, sometimes it's really hard to tell which color is which. Another problem is that on the microscope you have this optical crowding issue because each dot is diffraction limited, so you can only fish a limited number of dots in the cells. So how do we overcome those issues and to apply the fish to apply fish in the single cells level? And so this is what I'm trying to talk. I want to convince you guys with the rest of like 15 minutes. This problem could be solved or not be solved, could be like overcome in the sequential fish method. This method has been developed by Dr. Long Tsai's group at Caltech for the last decade, which Dr. Tsai is the student of the pioneer in the group, Dr. Shi at Harvard. This idea is that for you to this idea is that you can target, in this case, just liberate for the messenger RNA. We can imagine also this messenger RNA could be replaced to all kinds of other molecules. You can also hybridize against the same molecule multiple times with the imaging and stripping in between those hybridizations. For example, you have this mRNA 1 here. This is paint green at the first time, and then on the second round of hybridization, the color is switched to blue, and so on over the end time of the hybridization. You can generate a barcode, a temporal barcode across multiple rounds, and this allows you to distinguish between the different slices. So if you start with 5 colors, just 3 rounds of hybridization, it will give you 125 barcodes. This is really easy math, right? But over 6 rounds, it will generate around 15,625 barcodes. And this is going to allow you to identify hundreds of different molecules in the single cells level. So after the first method paper, Nature method paper published back in the 2014, they showed that SeqFISH could work well at the transcription level. In here on the left side figure, I'll also want to give a little bit background knowledge about this, about the introns and how they applied it here. So the pioneer work on the single molecular fish observed the nascent mRNA are produced in bursts at transcription active sites in individual nuclei. Particularly this nascent sites of transcription near the genomic loci could be selectively labeled over mature transcription by targeting the introns. And the introns, as we all learn, is going to be processed out. So when you fish against those introns, what do you say on the right side here is what you say is just only the nascent cytogenesis. And on the right side, you can see that nuclear and each of the dots is a single transcription active site that happen to be transcription at that moment. And what we're looking is we're interacting through the different chromosome, and most of the time you can see the two allies, and you have other nuclear structure like this, some of the DAPI rich regions in the cells. But in some sense, we got away with those two difficulty or two major challenge we were talking about, because not all of the genes are like active or are active like in that given moment. So around like maybe 20,000 genes, there's only like 2,000 to 3,000 genes are actually active or are in the bursting stage. So this is lower the number of dots in the cells. The dots in the cells. So in here, this the SeqFISH has been applied in the NIH 3 cells. This is our demonstration of how this works. And on the left side, this gives you a general idea of what's the fixture like. What's the image from the like microscope actually look like. And this is what I'm talking. That when you actually look at the image, it's pretty hard to tell which color is which. Which color is which in this like such a dense image. So when we look on the right, and we can see that like where is the individual transcript are, and this is being done on the mRNA level, which is more highly expressed in the cells. Probably like for mRNA, we're talking about hundreds of thousands of even like millions of transcript total in one single cells. And in here of the SeqFISH, we are able to do around like 10,000 genes, and this is like about like 50,000 barcode in just one single cell slice. But if you integrate them all together, all the slice, and average them out, this is gonna get you around 500,000 of transcript in a cell, which is pretty good number for the transcript. And in here, SeqFISH is already like a pretty good, like pretty impressive tool, but it is pretty hard to apply this to like any level of involved, like more than like 10,000, like more than like 10,000s of the size you want to target, due to the optical density of the transcript like in the cells, and as I mentioned, each mRNA or each like dots occupies a diffraction limit spot in the image. So there are 10,000 of the dots per cell depending on the cell types. So the optical crowding issue like actually prevent this technology to be applied to the mRNA or like the DNA levels. So Dr. Ted's group upgrade this method to like a method called SeqFISH Plus. So in this method, you can achieve super resolution imaging and multiplexing of 10,000 genes in a single cell using sequential hybridization and with a standard confocal microscope. At the SeqFISH level, there were only like 4 or 5 colors, but at the SeqFISH Plus, you can use up to like 60 pseudo colors. They are used in the 3 fluorine channel and to generate barcode within each channel. This is going to be really helpful to avoid the chromatic aberrations between the channels. In this way, repeating the pseudo colors 4 times, and we can use 1 time for the error correction. You can achieve easily 24,000 genes in single cell level. And the down graph show you that compare also with SeqFISH, the SeqFISH Plus with 60 pseudo colors is 8 times faster in the imaging time. With a SeqFISH Plus, is a way to do deterministic super resolution microscope. There are different other ways to do super resolution. One is using the traditional blinking, like palm and storm mechanism, where you have a single flow cord that blink on and blink off. You could do super resolution that way, but the problem is that you have to detect a single dye moleculars, which is seems pretty easy, but it's actually very hard to achieve in the single cells. Probably the one time you can get away is when you use it in really fly cells, so you can apply this to tissue because there are autofluorescence background. It's just going to be really hard to actually apply this into the day-to-day science. Another way to do is using expansion microscope, which was how this can be done back in the first methods. But when you expand the samples, you have to image longer, and this also bring one of the problem is that the sample will continue to morph. So you can repeat around hybridization and make it hard. This is going to make it really hard to line the dots over the multiple rounds. So by using multiple rounds of hybridization, you can target the subset of a very densely populated area. So you have this region with many RNAs, and if you image all those together in a diffraction limit microscope, you will see that those dots going to be overlap with each other. But if you use sequential rounds, if you use sequential rounds of dye to call only hive to only call a subset of them, then you can Gaussian fade the dots to shrink their size and irritate through the different dots in this little cluster. Then you can reconstruct a super resolved image, right? At the way higher resolution, where the dots are not overlapping. This could be repeatedly over 3 or 4 rounds, and you can start barcoding the transcript. For example, this color show at number 16 as a pseudo color, and on the first barcode, you could see you could decode exactly which gene is because this is how you design the DNA probes. That way, they spell out to this particular barcode. So this is how they could do the experiment with 10,000 genes. And you can think about this way of doing super resolution microscope, because you can control which molecular is turned on and turned off. It happens deterministically, and each time a dot lights up, it made of tens of dyes molecular because you have many dyes on the origin moleculars. So in this way, you increase the signal levels, and you guarantee you can detect something over time. And the next figures show that the RNA SeqFISH could be applied in the tissue level. In here is in the mouse cortex. So in here they can image around 10,000 genes in the tissues. We can zoom in from the very left side and to the very right side, and then down. So we can zoom into the regions of different regions of brain. In here, we focus on the mouse brain cortex, and we zoom in further, we down to the cells level. You can see this sort of like those kind of messy or like really dense dots of this 10,000 different transcript. And then when you zoom in further, you can see the location of each transcript with about like 100 nanometers resolution. With all those previous introduction, now we can talk about like apply this approach to looking at the chromosome in the cells. And we can do this in a way that the same cells, we can measure the DNA with SeqFISH, and we are looking at about like 70 different messenger RNA as well as the introns. And then we use the DNA fish to target the thousands of loci. Particularly, we target the loci around 1 MB resolution across the genome. So that's about like 2,000 loci overall, and this is done in the most embryo stem cells. Then each chromosome we can target about 60 regions, and there are 25 kilobase part. So this is much higher resolution within each chromosome. And after all this is done, we can do immunofluorescence on about 17 antibodies for different epigenetic mark, as well as nuclear body mark in the same cell. So this way we can interact all those together instead of doing RNA-seq, DNA-seq, or the DNA fish on some certain epigenetic mark separately. And here is the one point I want to touch before the end of time. So if you look at just the global level of those epigenetic marks, the global level is really different in the individual cells. So you can see that H3K accelerations, the cells right has really different global staining versus the other cells, and for different marks, it's also different. And this is definitely not a staining artifact. This could be show like very reproducible. And not only, and on the right side, this is going to be the kind of similar information showing the heat map group, and we can actually more directly see the variance in the different cell clusters and in different epigenetic mark. So before, all we do is we get a bunch of cells, we cross-link them, and we get the informations, but it's like in the cell populations wise, and we choose to, and because technology limitation, we can able to go down to the single cell level. But now, when we could measure the single cell, like in the expression level, this is going to be like review like more information and give us more chance to find out what is happening in that level. But not only we can measure the expression level in the single cell, we can also measure how long those like heterogenetics in the global epigenetic state lasts. And due to the time limitation, I can't go to detail too much. Okay, so now let me just stop here and start summarize what I have been talking about for the past 22 minutes. The take-home message that the many now repetitive like DNA loci and the size appear on the surface of the nuclear bodies. So this is what we've been like thought for the past time. And with this new technology, the picture of the nuclear maybe it's not like the spaghetti and meatball type. It's more like inside of the baguette bread, there's like a bunch of different holes, and both sides they're actually appearing on the surface. They're made of probably inside a lot of proteins and also like repetitive like DNA oftentimes in the nuclear bodies. And then when you put those together, you can see that the interaction between the DNA loci and nuclear bodies actually impose a very deterministic structure on the nuclear. And this is not really random. And there's a very constrained diversity of the possible state of nuclear can exist in, and also there's the long life states with the last slide I've been just briefly mentioned. So this technology overall is like a really impressive technology. This could reveal the information we have not able to detect for the past decades of research, and this is going to bring like a new revolution to the currently molecular biology. And I thought this is really cool, and so I want to share it with you guys. And if you have any questions, I believe Dr. Mayer said there are going to be like question section, and I'll be happy to answer some of the questions. Thank you guys.